सादर प्रणाम। एनसीईआरटी की वेबिनार सीरीज़ लिसनर टू लर्न और सुने और सीखें, इस श्रेणी में आपका एक बार फिर से स्वागत है। व्यूअर्स, आज का ये प्रोग्राम एनसीईआरटी के पीएम ई विद्या चैनल्स पर 9, 10, 11, 12 इन चैनल्स पर लाइव प्रसारित हो रहा है। साथ में ये प्रोग्राम आप यूट्यूब एनसीईआरटी ऑफिशियल यूट्यूब चैनल पर देख सकते हैं। आप अपने प्रश्न हमारे जो स्क्रीन पर दिखाए जा रहे हैं ईमेल एड्रेसेस पर भेज सकते हैं, टेलीफोन नंबर पर भेज सकते हैं और साथ में यूट्यूब में भी पूछ सकते हैं। दोस्तों, आज का ये प्रोग्राम हमारे लिए बड़ा मुख्य है कि आज हम अपने एक महान वैज्ञानिक, भारतीय मूल के महान वैज्ञानिक डॉक्टर हर गोबिंद खुराना के संदर्भ में कर रहे हैं। और आज इस चर्चा को करने के लिए हमारे साथ ऑस्टिन, टेक्सास, यूएसए से प्रोफेसर सहोत्रा सरकार शामिल हुए हैं। प्रोफेसर सरकार हम लोगों को हर गोबिंद खुराना साहब की ज़िंदगी के बारे में, लाइफ के बारे में विस्तार से आज हमारे साथ 1 घंटा चर्चा करेंगे, हमें बताएंगे। यहाँ स्टूडियो में मेरे साथ प्रोफेसर सुनीता फरकिया मेरे साथ हैं, और जो कि हमारे विभाग विज्ञान तथा गणित शिक्षा विभाग की अध्यक्षा हैं। वो आप सबके साथ रूबरू होंगी और प्रोफेसर सरकार को आपसे मिलाएंगी। आज का ये प्रोग्राम है, आज की टॉक में, आज के उस उद्बोधन में मेनली दिस इज ए बायोग्राफिकल स्केच ऑफ डॉक्टर हर गोबिंद खुराना। Friends, while being a faculty member at University of Wisconsin-Madison, United States, Dr. Khorana shared the 1968 Nobel Prize of Philosophy, which is also considered the Nobel Prize of Medicine, with Marshal Nirenberg and Robert Holley for research that showed the order of nucleotides in nucleic acids. It is understood that this nucleic acid carry the genetic code of the cells and controls the cell synthesis of proteins. 21, this is the November 21, this is the birth centenary year of Professor Karana, an Indian American biochemist, Nobel laureate. Friends, in the same series we will be having another lecture on 26th of November on the Nobel Prize, on the institutions of Nobel Prizes. Dr. Sarkar will also be speaking about the genome connection with the CRISPR technology a bit, and on CRISPR we will be having another talk on 26th of December by Abhishek Mishra, who has worked with the Nobel laureate Carpenter, the Nobel Prize which was given in 2020. Now I request Dr. Sunita Farakia to kindly welcome Professor Sarkar and introduce Professor Sarkar to all of you viewers. Dr. Farakia, please.
Good morning and warm welcome, viewers. Today I am feeling great pleasure and I am feeling honored to introduce today's speaker, Dr. Sahotra Sarkar, who is a professor in the Department of Philosophy and the Department of Integrative Biology at the University of Texas at Austin, USA. Originally, Dr. Sarkar is from Darjeeling, India, and he was educated in Columbia University and University of Chicago. He specializes in the history and philosophy of biology, environmental philosophy and conservation science. He has held fellowships at MIT, Institute for Content Studies at Berlin, the Max Planck Institute for History of Science and Edelstein Center for the Philosophy of Science at Hebrew University, besides teaching at McGill University. He is the author of 7 books and the most recent being Cut and Paste genetics. A. CRISPR Revolution 2021. So, I am we are fortunate enough, Dr. Sarkar, that you are with us today, and I welcome once again you to this forum and our viewers also. Thank you so much, sir.
Yes, so friends, this month is important for all of us Indians. We had on 7th of November, we had the birth anniversary of our first Nobel laureate in science, C.V. Raman, and yesterday we had National Education Day, the birth year of 134th birth year of Maulana Abul Kalam Azad. So, Professor Sarkar, it's all yours now, please.
Thank you, everyone, and thank you very much for asking me to give this talk and hosting me. So I will be looking today at the life of the first Indian American to get a Nobel Prize, Har Gobind Khorana, who got the Nobel Prize, as was mentioned, in 1968. And I will talk about his early career, the work that made him mainly famous, that is the work on the genetic code. Then I will talk about his later science, how he tried to synthesize, or succeeded in synthesizing a complete gene, and then how he worked on membranes for the last decades. But I'll mainly be focusing on the genetic code and his work on that, the decipherment of the genetic code that led to his Nobel Prize. Now, Kurana did not know the date of his birth. Official documents gave the date as the 9th of January 1922, but no one knows where the date came from. Kurana came from a very poor family in Punjab, what was Western Punjab then, in 1922 part of British India, and now it's part of Pakistan. He was 6 years old before he received his first pencil, which was a birthday gift from his father. But his father, to his horror, he got very sorry that the pencil actually broke into two when he tried to use it, and it left him crying. But the father comforted him, saying he could sharpen both pieces of the pencil now, and he could have two pencils. And yet this boy from Punjab in 1968 shared a Nobel Prize for physiology or medicine with Marshal Nirenberg, who, along with Karana, independently solved the genetic code, and with Robert Holley, who sequenced the first transfer RNA molecules. These are the molecules that are used by ribosomes in cells to construct a protein using the information in the messenger RNA, which gets that from your genes, that is the DNA. Kurana's Nobel Prize, here is a picture of that. They're all in Stockholm, and that's Holley, and that's Nirenberg, and that's Kurana. And they are, we do not know what they were talking about in this picture, but I would, given that it was Har Gobind Khorana, I'm sure that it was science. So from an impoverished village in Punjab to a Nobel Prize, it's an incredible journey. And our story today is of that journey. At the time when Kurana solved the genetic code, he was only 44. He was 46 when he received the Nobel Prize. He had 4 decades of science left in him, and there were a lot of other brilliant achievements that occurred during those 4 decades, including the first synthesis of a complete functional gene. That was a gene that coded for a transfer RNA, and Kurana succeeded in putting it into a bacterium and showed that it was functional. Those stories I will leave for a different day. I will say a few things about it at the end, but I'll focus on the first part of his journey. So he was born to a Hindu family in a village called Raipur that had only about 100 people. It's near Multan in Punjab and now in Pakistan. The father, Ganpat Rai Karana, was a Patwari, and he was poor, but he was dedicated to educating his children, and the family was practically the only literate family in the village. His mother was Krishna Devi Karana. She was a homemaker, kept their home. There were 5 siblings, 4 sons and a daughter. Gobind was the youngest of them. There are many anecdotes of the family's poverty. I already told you about the pencil. There's another story that when he was young, he would wake up very early to go out into the village and look for a house with smoke coming out from the chimney and ask for a bit of ember, a small piece of coal that was still lit, to take home to light the cooking fire. There's also the story that when he was in school, he wanted to join the Boy Scouts, which is what has become the Bharat Scouts and Guides today. and the family could not afford the uniform. So his mother took old pieces of cloth from different areas and sewed together a uniform for him. That was not good enough for the scoutmaster. When Kurana showed up wearing that uniform, he was not allowed to join the troop. However, Ganpat Rai taught Kurana to read, Gobind to read very, very early. I'm sometimes going to call him Gobind because that was the name by which he was known all through his later life. When I met him at MIT, the first thing he told me, call me Gobind, everybody calls me Gobind. His first few years of schooling took place under a village tree, which was not unusual for India at the time. But the village was lucky enough to have a one-room school that his father had helped establish, and that's where his education continued. And because he knew how to read and write, he had an important function to play when he was in the village. He would sit on the steps of a post office and write out letters for all the townspeople, most of whom could not write at all. It was then that he developed a very characteristic, very precise microscript handwriting, which anyone who ever worked with him would instantly recognize. He also developed his trademark frugality. He would use his lead pencils until he could almost not hold them, and would take notes on any scrap of paper that happened to be available. And he would often say that he did exactly what Gandhi used to do when he too used to write. The backs of envelopes were as important as bought and important expensive pieces of paper. Ganpat Rai, his father, was adamant that his children's schooling did not end at the village. So Kurana attended the DAV High School in Multan. This was founded by the Dayanand Anglo-Vedic Education Society, which propagated a Hindu fundamentalist ideology and founded the proselytizing outfit Arya Samaj. The first school was established in Lahore in 1885. The one in Multan, the one that Kurana attended, was actually established in 1896. We don't know much about his school days. It did not affect him very strongly. He was strictly secular in his later days and worked with anybody and everybody, irrespective of any religious or other background. The only thing he recalled from the school in later on is that there was one teacher called Ratan Lal who influenced him very strongly during his early years. School was followed by college at the University of Punjab. He had developed a liking for English literature, particularly the works of Bernard Shaw. In fact, it is sort of a funny story about how he ended up being a chemist. And I am going to read from his own words. At age 18, when I sought admission to the Punjab University in Lahore, I applied to two departments, English literature and the honors course in chemistry. Admission in the latter was restricted and required an interview. My shyness kept me away from the interview, but they saved a place for me anyway, and thus I became a chemist. We wonder what would have happened if he had actually studied English. At Punjab University, Karana continued his studies to obtain an MSc in chemistry in 1945. His supervisor was someone called Mahan Singh, whom he called a great teacher and accurate experimentalist. That's where he published his first two papers, both of which came out in the proceedings of the Lahore Philosophical Society. These papers are rarities. They're very difficult to find now, but they were excellent papers. His early career, at the end of World War II, in 1945, the British government of India initiated a scheme for scientific and technological development. They already knew by then that a decision had been taken that at some point in the near future, India would become independent. So every year, a large number of Indian students were selected for training abroad on scientific and technological areas. Kurana was in the first group selected in 1945. He was selected by the Ministry of Agriculture, and he was assigned to an institute in Berkshire, in England, to train using insecticides and fungicides for agricultural development. He was supposed to bring all of this knowledge back with him and use it in India to increase the efficiency of food production in our fields. The Indian High Commission in London was supposed to secure the position in the institute in Berkshire for him. So Kurana arrived in London expecting to go to Berkshire, but then he found out that the High Commission had not been able to secure the position for him. At the end of the war, British institutions were overcrowded from the return of ex-soldiers who wanted to continue with their training, and so a place was not available for him. So the High Commission, without being able to consult Kurana, looked at his background and found that it was in organic chemistry. And so they got a place for him. a regular PhD studentship in organic chemistry at the University of Liverpool. And he was assigned Roger J.S. Beer as a PhD supervisor. Beer was a very good scientist who had just received his PhD from Oxford. He was a very good supervisor. Karana later said, the association with Roger Beer proved to be most rewarding. Not only did he supervise my research, but he otherwise taught me a good deal. The research consisted of the synthesis of organic compounds that were called indo-alkaloids. These are molecules that have medical significance. They often affect the nervous system as well as melanin, which is the molecule that gives you the color of your skin. Karana was awarded a PhD in 1948, after which he was supposed to return to India under the terms of his scholarship. But he wanted a postdoctoral year to stay in Europe, and the official story was he very much wanted to spend a period of time in the laboratory in a German-speaking region of Europe. The reason was that he desired to be able to read German fluently, because most of the important science of the period, particularly organic chemistry, was done in German. It was all published in German. But he did so, he wanted to be in a German-speaking country, mainly because he thought he wanted to go to Switzerland, which was the only German-speaking area in Europe which could take students at the time. Germany and Austria, after having lost the Second World War, were in no position to do anything of that kind. So Karana wanted to work with a scientist called Vladimir Prelog. He was a Yugoslav who had actually managed to escape after German occupation and had ended up in the Hoch School in Zurich in Switzerland. And he was supposed to be a legendary mentor and a great teacher. And Karana wanted to be able to work with him and learn from him. But there was also an unofficial story as to why he wanted to go to Switzerland. In 1947, while he was in Liverpool, he had gone to Prague in Czechoslovakia to attend the first World Festival of Youth and Students. These were festivals that were organized by the Soviet Union, partly as propaganda, but partly also to establish a network of students around the world who would work for peace and harmony. And at that particular festival, he met a young woman by the name of Esther Elizabeth Silver. She was Swiss and was attending the festival with her parents. Esther was interested in having an Indian pen friend, and Kurana thought about his own sister and thought that she would be suitable. But he clearly was attracted to the woman, and he traveled to Switzerland to hike with her brother. And then he stayed in touch with Esther Silver, and that probably gives the best explanation of why he wanted to go to Switzerland so badly. So he applied for money with the government of India for a year's postdoctoral training. But in 1948, this is the year after independence, the new government of India was practically bankrupt. It could not provide for students returning from abroad, for them they could not provide jobs, and they actually terminated the scheme, the scholarship scheme, by which Karana had gone to Europe. You have to remember that India was drained of all its resources given by the British to fight the Second World War. A lot of, not only were a lot of farm products, especially rice, taken from India, but the treasury was completely depleted, and Nehru, as prime minister, essentially was trying to govern the country without a budget. So it's not surprising that Karana's application for funds for a postdoctoral stay was denied. But he decided to go work with Prelog anyway. He had some savings left from the money that he had from his scholarship at Liverpool, and he convinced the Indian High Commission in London to assist him in get to Zurich and postpone his return to India for a year. And so he arrived in Prelog's lab unannounced. He had no letters of references, no letters of introduction. All he had was a thesis. And all he wanted, he said, was some space to do experiments. He did not even expect to be paid. Prelog, who was legendary in being fair to people, took the thesis, told Kurana to go away and come back after a few hours. Prelog sat at his desk and read Kurana's thesis, and he immediately accepted Har Gobind Khorana as the student, even though there was no money to pay him at all. For 11 months, Kurana just lived in Zurich, and he worked in that lab, and presumably he spent some time with Esther. And Sakmar, his postdoc, later said he essentially lived in the laboratory on rice and unpasteurized milk, the cheapest milk that he could buy from farmers in Switzerland. Prelog was actually a legendary mentor. He would eventually get his work for, he would get a Nobel Prize for his work on stereochemistry in 1975. And with Prelog, Kurana worked on the chemical structure of certain alkaloids which are active in the poison arrows, the poisoned arrows by which Native Americans hunt in Brazil, in Central America, and elsewhere in the Americas. And Kurana did some very good structural work on that, which got published. And more importantly, he developed a collaboration with a scientist by the name of George Kenner, who was also working on the same alkaloids and had come to work with Prelog from the University of Cambridge. What's even more important is that besides working in the laboratory, during this entire period, Kurana was doing exactly what he said he would be doing. He was learning how to read and write German completely fluently. And so he would sit down in the lab and he would get scientific literature in German. And not only would he read them, he would try to translate them into English. And in the process, he came across a series of papers by a man by the name of Fritz Zetsky, who had been working on some very little known chemicals that were called carbonymides. And one of them is called the DCC, the dicyclohexylcarbonamide, which nobody ever says. It's too complicated to pronounce. And it's very, very simple. It's two carbon benzene rings, and from both of them, a nitrogen atom hangs, and the nitrogen atom is double bonded. Each of the nitrogen atoms is double bonded to a carbon atom, so the carbon has a valency of 4, nitrogen has a valency of 3, and you have the benzene rings here. And what is wonderful about this molecule, as we will see later on in the story, is that this is the reagent that cracked the genetic code. So after the time in Switzerland, Kurana had to return to India, which he did. India was now independent but partitioned into two countries, India and Pakistan, and the promised government job that he was supposed to return to as part of his scholarship simply was not there. Punjab University, my alma mater, was now in Pakistan, and many of my friends who had previously been at the Punjab University were now unemployed. He lived for several months in the servants' quarters of his uncle's house in New Delhi. And in fact, many of his old professors and his old friends who had been students at the Punjab University were wandering around as refugees after the great population exchange with absolutely no job. So Kurana, luckily, given the circumstances, the government of India said that Kurana would not have to pay back the money of his scholarship, and he could do whatever he wanted. And he had remained in touch with George Kenner, who had gone back to Cambridge. Thanks to Kenner's influence, Kurana got a postdoctoral fellowship to work with Professor Alexander Robert Todd, who also was awarded a Nobel Prize in 1957. Somehow or the other, Har Gobind Khorana managed to work with some of the best mentors in the world in his day. And Kurana was actually supposed to work on peptides that were related to a new hormone that had just been discovered, and his entire extended family in Delhi scraped together enough money for his fare back to Britain, and he went back after a few months in 1949. Ever since going back to Britain that time, until the end of his life, Kurana came to India only twice. Because of partition, he had lost almost all his personal connections with India, and he developed connections elsewhere, and never actually managed to have a stable connection with his ex-family here. In a way, he was lucky that he got to Cambridge. As he put it, Cambridge was the place where the new molecular biology was being founded. It was a uniquely exciting place at the time. Todd's own work was at the climactic period of defining the chemical structure of nucleic acid. Frederick Sanger was sequencing insulin, which was the first protein to be so tackled. At the Cavendish Laboratory, Max Perutz and John Kendrew were embarking on the first X-ray structures of myoglobin and hemoglobin. And soon Watson-Crick structure for DNA, that's the double helix model, would emerge from the same laboratory. Molecular biology was in the making. It was exciting to be in Cambridge. This stay broadened my intellectual horizons and was of decisive value in my scientific development. Karana was involved in many different projects at Cambridge, including work related to the new hormone that we just talked about. More importantly, with working with Kenner, with George Kenner, he began developing techniques for the synthesis and stepwise degradation of peptides, that is, taking peptides apart. Har Gobind that he had first encountered in Krylov's laboratory turned out to be useful for cleaving off amino acids from peptides, that is, cutting them into smaller pieces. But it turned out to be not of that much value because of too many side reactions that were happening. But then he also noticed that DCC was a useful reagent to synthesize DNA nucleotides and nucleosides together. And in particular, it could be used to synthesize, make from scratch pyrophosphates. And this is what will turn out to be the crucial thing that he would know when it came to deciphering the genetic code. At the end of 3 years, his fellowship ran out, and this is how he explained it. I was looking for a job virtually anywhere in any country. Gordon M. Shroom of the University of British Columbia, UBC, Canada, whose phenomenal energy allowed him to hold several administrative positions simultaneously, had also taken on the job of the directorship of the British Columbia Research Council, Vancouver, that year. The British Columbia Research Council was a very small organization that was set up by the British Columbia government. It was located at the UBC campus and carried out research to aid the local industry. Gordon Trump was visiting the UK and other countries to recruit people for UBC for the Research Council as well. I was interviewed by him. I didn't know where Vancouver was. To go there if Dr. Shrum would offer me a job. Gordon Shrum had obtained a small grant from Steve C., a well-known physical chemist who was president of the National Research Council, that is of Canada, in Ottawa, and wanted to see some fundamental research started there. Gordon Trump, who is a physicist by training, also told me quite candidly he was looking for an organic chemistry because he thought that organic chemical research was the cheapest to carry out, requiring only test tubes. Kurana was actually absolutely delighted. For the first time, he would have his own laboratory. And while the funds were limited, he had all the freedom in the world to do what he liked. Esther and Karana got married in Switzerland in 1952. Karana left for UBC immediately afterwards. He went on to Vancouver. Esther followed him there a few months later. They lived there until 1960. All three of their children were born there. Julia Elizabeth, the oldest, whom I've been interviewing, and who told me most of the stories that I know about Karana. Emily Ann, who died in 1979, and Dave Roy, who was born in 1958 and whom I've never met. Karana in Vancouver continued to focus on the carbonimides, the special reagents that he had come across when he was in Switzerland. And he pursued a program of synthesizing sugar phosphates and biochemical inputs. And this is where his greatness as a scientist begins to emerge. He succeeded in synthesizing from scratch ATP. That is the molecule that gives you all the energy in your cells. And also ADP. ATP is adenosine triphosphate. ADP is adenosine diphosphate. And the reagent that helped him do all of this was DCC. And he was sold in it. In fact, the work was so important that even though he had been relatively unknown until that point, and was still very, very young, very prominent biochemists began to visit his laboratory to learn his techniques. These included Paul Berg and Arthur Kornberg, who both went on to win Nobel Prizes. And Kurana began to see that his organic chemistry could be extended into biochemistry, and there was very important research to be done over there. So he actually visited Berg and Kornberg in their laboratory in St. Louis, Missouri for a few weeks and learned their techniques too. And the research began to undergo a transition from pure organic chemistry to biochemistry. By 1960, he had finished his first major project. He had from scratch carried out what he called the total synthesis of coenzyme A. This was the first large biologically relevant molecule that had ever been synthesized outside a living system. And Kurana had succeeded in doing this at Vancouver. And after he does it, he just announces. Moffat, who's the co-author of the paper, was his first ever graduate student. The synthesis of coenzyme A and the work reported in the accompanying papers brings to a close the program of research initiated in this laboratory some 7 years ago on the development of methods for the synthesis of unsymmetrical pyrophosphates of biological interest. The methods now available are believed to be satisfactory and completely general, so that the synthesis of any nucleotide coenzyme or related pyrophosphate may be undertaken in the laboratory with confidence. He had shown that these large molecules could be made there. DCC also allowed the synthesis of DNA, both stepwise, when you take a string of DNA and add one cell at a time, and also by the polymerization of nucleotides, where entire groups of residues of DNA get added to it at the same time. This resulted in a very ambitious project by the end of the 1950s. Kurana wanted to synthesize an entire gene. Genes were known to be DNA by now. They were known to have strings of nucleotides. And Kurana wanted to put an entire gene together in the laboratory and then transfer it into a living organism and make sure that it functions. It would take him 10 years, okay? And he had absolutely never wavered from the project. And as early as 1960, he announced it. In the distant future, the total chemical synthesis of macromolecules possessing biological function must also be considered. The problems are vastly more complex than anything previously undertaken by experimental organic chemistry. Nevertheless, biology urgently asks, will organic chemistry extend its horizons and accept the challenge? And then in the middle of all of this, as almost a detour, a side trip, he solved the genetic code. In 1960, Karana moved his laboratory from Vancouver to the University of Wisconsin-Madison. From Vancouver, after synthesizing ATP, he had written to Van Potter, a leading cancer biologist at Madison, asking if he could test his synthetic ATP in rigorous biochemical assays. Van Potter not only obliged with the experiments, but also succeeded in bringing Gobind to the Institute for Enzyme Research, a vanguard of chemical research at Madison. From 1960 to 1970, Gobind was co-director of the Enzyme Institute and member of the Department of Biochemistry at University of Wisconsin-Madison. This is where he did his Nobel Prize winning work on the genetic code, and he also got the Nobel Prize in 1968. And here is a picture of his group in front of the Institute, all of them working on these problems that he's done, and Kurana himself is right in the middle of the group, right over here. And this is T.M. Jacob, who had come from India to work with Kurana in the laboratory. Jacob is preparing to carry out an assay that will show that nucleotides would be sequenced in the way they were intended to. Now recall that Kurana was at Cambridge. I just mentioned that a few minutes ago. At the time when the molecular revolution in biology was beginning, just after he left, Watson and Crick proposed the double helix model of DNA. In the double helix, there could be any sequence of nucleotide bases, A, C, T, or G along the backbone, and then base pair complementarity set in. A was always paired with T, C is always paired with G, and that determined the structure. Combinatoric spread specified gene identity and difference. What exactly is the sequence in a string of DNA determines what gene the DNA is. But genes also specify proteins, and proteins also had a linear sequence of amino acid residues. The relation was one of coding, and as early as 1941, Erwin Schrödinger, one of the founders of quantum mechanics, had speculated and predicted that a code would be found, and he called it the hereditary code script, that it would be found in living organisms. Crick, shortly after coming up with the double helix model for DNA, embedded this idea into the idea of biological information, saying that what DNA carried was information that specified what the protein is. And to understand what this information would be, what you had to do is look at the coding relationship between DNA and protein. And a lot of people, Crick, the physicist Gamow, he's the person who came up with the Big Bang theory of the universe, they all began to develop speculative models of what the genetic code would look like. But what was the code? It turns out experimental results showed that all these theoretical speculations of what the coding relationship between DNA and protein was completely wrong. Instead of theorizing, what the scientists should have been doing were experiments in the laboratory. And that's what two people, Marshal Nirenberg and Holley did. What they did was they developed a cell-free system. You do this by taking a cell, okay, cleaving its membrane and making everything from inside the cell go out into the test tube. You no longer have a living cell, but you have a system without the cell that can do a lot of the things the cell does. And one of the things that Nirenberg did was it took a sequence of nucleotides, which is the RNA that comes from the DNA, and he dumped it in it. And the sequence it started out with was all uracil use, which in RNA corresponds to the thymines of T in DNA. And the amino acid residues that was coded by them, the peptide that came out, consisted entirely of phenylalanine residues. And so you suddenly knew that U coded for a phenylalanine. And all the theoretical speculations about the genetic code turned out to be wrong. The code would have to be experimentally decided. And so a race was on. What was the code? How will you defend it? How will you discover it and show it to be correct? The effect on Har Gobind Khorana was electrifying. DCC allowed him to create virtually any sequence of DNA nucleotides that he needed. Since the code was triplet and you had 4 different nucleotides, the total number of possibilities of triplets called codons was only 64. You get the number by multiplying 4 to itself. So in 2 places you get 16. You multiply that by 4 because it's a triplet, and you end up with 64. And Kurana knew that he could create all 64 of these triplets and put them inside the cell-free system and look at what polypeptide came out, and there you have it. You have solved the genetic code. There was competition between the laboratories of Karana, Nirenberg, and another Nobel laureate, also Ochoa. Nirenberg was hampered by the set of nucleotide sequences he could construct. He did not have Kurana's technique using DCC. Kurana was not restricted. Gobind's group in Madison worked around the clock in double shifts to synthesize all possible trinucleotides, that is the triplets, thus providing a firm basis to establish the complete codon assignments and to determine how the code was read. Crick, who had been dabbling with experiments, was also very excited about Kurana's work, and he repeatedly visited Madison, encouraging Kurana to do this. By 1966, the code was complete, and in 1968, Kurana and Nirenberg were awarded the Nobel Prize. And here is a picture of all the Nobel Prize winners from 1968. From left, you have Kurana and Holley, who shared the chemistry or physiology or medicine prize together. Then you have Luis Alvarez, who's the one who eventually discovered that a meteorite striking the Earth had killed off all the dinosaurs. He got the Nobel Prize for physics. Marshal Nirenberg got the Nobel Prize with Kurana. Lars Onsager, he also got the Nobel Prize for physics because of his work on thermodynamics. And then the Nobel Prize for literature went to Yasunari Kawabata, who's on the right. And that marks the end of the most successful period of Kurana's entire career. In 1970, Kurana moved his laboratory to MIT, and he remained there for the rest of his life. That's where I met him. Here he started work on DNA amplification, and that eventually led to PCR. But Kurana, in the meantime, had moved on to do work on membranes. Even more importantly, he completed a project which he first envisioned over a decade earlier when he was still in Vancouver. And this project was the total synthesis of a functional gene. In 1972, he synthesized a suppressor tRNA gene and showed that the synthesized gene was functional in a bacteria. This, more than anything else, is the beginning of what we now know as synthetic biology, where by ordinary chemical mechanisms, you artificially make something and show that it has all the biological function you would want from it on just the basis of its construction. Another way of putting it is that while nobody has done it yet in the laboratory, this is the beginning of, it must be the beginning of any project that would actually create life from the laboratory. Kurana, after synthesizing the gene itself, Kurana himself decided that he actually wanted to move on into other directions and do other things, and he explained that. in the mid-1970s. Nucleic acids had been my focus for more than 25 years. The work on the total synthesis of genes and their cloning had still to be completed, but the strategies seemed clear. While I was not about to say farewell to nucleic acids, the idea of starting anew in an entirely new field began to take hold in the early 1970s. I began to think about biological membranes with a distant hope that I might get into areas of molecular neurobiology and signal transduction. As in my previous work, I did not believe that I would be able to formulate at the outset a specific area of major commitment. Rather, I hoped that clarity and specific objects would evolve in due course. Kurana's initial focus was on bacterial redoxin, a proton pump that transfers protons across membranes. Later, he shifted to rhodopsin. He was the first to sequence all of these, and he established all the basic rules of the chemistry and biochemistry of these molecules. The jury is out. One can wonder whether he achieved as much working with membranes as he had done with nucleic acid work. Here, there's a very clear analog between Karana and Francis Crick. Crick, too, in this period, turned away from DNA. So to say, all the low-hanging fruit had been taken. Everything that could be done relatively simply had been done by Crick, by the school, the French school in Paris, by Karana and people of that sort. And instead, they all moved in other directions. And both Crick and Karana began to be interested more and more and more in neurobiology and the human mind. Crick began to follow all kinds of studies of the human brain, hoping to bring functional neurobiology into the molecular realm. And Kurana thought that since a lot of the important instructions in the brain happened at the level of membranes and things that translated and things that involved membranes being connected to each other, for example, when you look at the dendrites of neurons and things of that sort, by focusing on those molecules that sat on the membranes and permitted signals to go back and forth into neurologically important cells, he might have a handle on neurobiology and the functioning of the brain to form the mind. Well, it did not really happen yet, but he realized by the end of his life that this would be a lot more difficult than anyone had expected, and so would Crick. They were basically on the same boat together. So what's his legacy? Well, as far as life goes, he continued membrane work for 30 years. Esther, his wife, died in 2001 from renal cancer, and pretty much marked the end of Kurana's productive care career. He lived on for 10 more years and died in 2011. We know very little about him as a person. Very little personal material is available either in print or in the archives. The story of his early life in India will never be known. Not much is known about his early relationship with Esther, his struggles and their love. What we do know is that he liked physical activity. He liked to swim. He swam almost every day. He liked to hike. It allowed unfettered thought. He was humble, and he was always willing to listen and to talk science with anyone. Uttam Rajabhandari, who worked with Kurana from 1962 onwards, first came as a graduate student, then went on to be a postdoc. In 1969, he left Madison to move to MIT, so he could spend a year setting up Kurana's laboratory. So the laboratory was already functional by the time he arrived in 1970. And then later on, he became a distinguished professor of biochemistry at MIT himself, and he and Kurana, when I knew them, had joint lab meetings together. He wrote in the obituary in Nature, Gobind used the solitude of hiking to think about scientific problems and always carrying a pen and note cards to write down his thoughts. On the day of his Nobel Prize announcement, he was among the last at the university to hear about it. As he often did, he had gone to a rented cottage by a lake outside Madison with no telephone or radio to write papers. His wife Esther had to drive over to give him the news. At the celebration, I remember him enthusiastically explaining his latest work on blackboard to two professors holding glasses of champagne. Gobind was at the heart an experimental scientist. With him gone, we lost an extraordinary man. And that's the picture that Rajabhandari wrote on the day that Kurana found out that he had won the Nobel Prize. The department brought a lot of champagne for them, but there is Gobind. He's drinking the champagne, but in the meantime, he's still explaining what his next experiments would happen. to two of his colleagues at the University of Wisconsin at Madison. What was his method? Kurana did not leave much of programmatic or speculative writing. He was as reticent about the philosophical motivations of his work, why he went about doing things exactly in the way he did, as he was about his own personal history. But he pushed 2 points. One of them was the idea that ordinary chemistry could tame biology. You carry out organic chemistry very, very well, and no matter how complicated the biochemical molecules are, eventually you'll be able to explain what it did on that basis. Something that philosophers call reductionism was absolutely fundamental to him. He did not like this new information-based picture of molecular biology. He realized very early that all this talk about information being in the genome rather than it being the chemistry of the genes, that all of this was metaphorical. And we know now that the informational talk in biology has not stood up to scrutiny. And the other part, of course, is even more important. This was the pursuit of total synthesis. First, he starts out with ATP. He synthesizes this molecule, which is the most important molecule providing energy to the cells. Outside cells, using ordinary laboratory chemicals, DCC was the most important of them. Then he goes on to make cofactor A, a complicated enzyme, total synthesis of that outside the laboratory. And finally, there was the total synthesis of the gene. It was almost a side trip to solve the genetic code. Given this entire progress of total synthesis, it was inevitable that he would have available to him all the methods that were required to solve the genetic code and all of that. In fact, Karana is the forerunner of the most important movement in molecular biology right now. Controversial because very few people believe that it will ultimately succeed. On the other hand, extremely well funded because all biomedical corporations want the techniques that are developed in this context, and that is what is called synthetic biology. I would end by thanking all the people that I've been interviewing and working with. Julia Karana, his oldest daughter, who's been giving me lots of personal information. I am in the process of writing a book on MCERT for all of you on Karana's life, which should come out some point next year on the occasion of his centenary. With centenary, there's Marvin Caruthers, who's a distinguished professor at the University of Colorado, Vinnie Cheroville, who went to graduate school with me at the University of Chicago, and now is on the faculty at IISc, Uttam Rajwandari, whom I mentioned earlier, who's at MIT, Thomas Sapmar, who's at the Rockefeller University, was also one of his postdocs, Dieter Zoll, who's done the most important work of the evolution of the genetic code since it was first decoded, and Matthew Jacob Tatsoville, who's also at IISc, whose father was one of Karana's postdocs. Let me stop here. Thank all of you for listening to me and wait for questions.
Thank you very much, Sarkar Sahib. You made us to learn much more about Kurana, and you covered his entire life, right from his childhood to his later days. And yes, there are many takeaways from his life. He remained active till his late 80s. You had given us the references of his work up till 2010 or so. Yes, indeed, magnificent. Just for the completeness, our viewers, the Dayanand Aglo-Vedic educational institutions, in its Multan school, where Kurana studied, it was later shifted to Jalandhar after independence. And in this birth centenary year of Kurana, this DAV organization is planning to facilitate Kurana once again and naming some portions of their schools in Delhi or in Jalandhar. So they are planning. So we will get back to you, Dr. Sarkar, for this development which is being planned by this DAV organization in India. I really do not know. We are not much aware that how much it is happening in the Pakistan portion of the greater India, where Kurana was born and studied. Yes, your life, your deliberation clearly mentioned and showed us that developments which were taking place at that time were not alone in the area of biology or in the area of chemistry, but there, yes, we know that many of the Nobel Prizes or many of the researchers in medicine have come from chemist sides. So, I have some certain observations for that. I will get back. I will just like to see your observations. However, you beautifully demonstrated that the Khorana was working with Schrödinger's understanding at that time in the quantum mechanics, the Gamows who was also at the Wisconsin at the same time, probably at the medicine itself, where he was developing his study with Weinberg at MIT on the grand unification theory, Big Bang theory of the universe. And you mentioned about the Moffat, who was working at around the same time on the behavior of materials under mechanical, electronic and other stimulus. At the same time, it's a beautiful connection of all the sciences, all the areas in sciences. So, but the question which comes to my mind, that what made Kurana to shift from organic chemistry to pursue in biology? Can you throw some light, Dr. Sarkar, on that particular shift, please?
So Kurana comes of intellectual age in the 1950s. That is the first time when people began to have sufficient knowledge of what biological materials are made up of to think in chemical terms. Now, for hundreds of years, people always kept on saying that biological things are ultimately chemical or ultimately physical and all of that, but that this just remained blues talk. There was no actual way of showing that. And then in the 1930s, after the study of enzymes becomes very, very commonplace, biochemistry emerges as a serious discipline, first at Cambridge and then elsewhere. And it was absolutely clear right then that biochemistry was in many ways applied organic chemistry. And then in the 1950s, when you began to see that large molecules that were important in living organisms, such as proteins and DNA, were also subject to biochemical rules and biochemical analysis, it naturally became plausible that you could study these using the methods of organic chemistry. But it was also very clear that it would be very, very difficult. And it is very typical of Kurana and his dedication to what he would do, that just because it was difficult, he did not avoid it. Because it was difficult, he took it upon himself to do these experiments, even if they would take 10 years to finish, and hundreds of people would have to be involved and collaborate. Over his life, he had more than 250 graduate students and postdocs. He really had the ability to force through a very well-organized lab to solve very difficult problems. And I think it's the love of the difficulty of using organic chemistry within biology with all its complexities that drove him in that direction, and in many ways separated him from, say, people like Crick or people like Watson or others who did not want to deal with that level of chemical complexity and just stuck to the level of the macromolecules themselves.
Yes, you also made us to remember that the pen friend, I mean, the huge dynamics of social behavior from pen friendship to the nowadays to the Facebook friendship. During this, all this, Khorana, who could live, who could give birth to an entirely new branch of biology that is called now synthetic biology, which is still a part of molecular biology. How do you think, Dr. Sarkar, that Khorana fits into the areas of or in the history of molecular biology? These questions are somewhat related to what I asked earlier, but it is probably in the same context. Yes, Dr. Sarkar.
It is a little difficult to answer, and one of them is that if you take classical molecular biology, the molecular biology of Crick, who was its leading theorist, really be biology rather than chemistry, because the concept of information was used. Central to it was the central dogma. Information resides in the protein, in the DNA, and it goes from DNA to DNA or DNA to protein. Once it gets into the protein, the information never goes back to DNA. That kind of reasoning did not impress Kurana. He was much more of a conventional chemist who just thought that without using the concept of information or any of these more controversial concepts, you could do everything you were supposed to do in the biological realm. And at one consequence of that, Kurana used to call his own discipline, what he created and what his students pursue, chemical biology rather than molecular biology. In 2000, he brought out a huge 700-page volume of his most important papers, on each of which he had given historical and interpretive comments, and he called it chemical biology. And all his students and postdocs, Rajbhandari, Sakmar, all of these people also use the term chemical biology for it. So I think the best way to look at it is, if you look at molecular biology broadly, there are multiple different strands within it. And one of them is the informational strand and all of that. And Kurana's chemical biology is a unique part of molecular biology, a unique school within it, which he almost single-handedly founded. And that's the one that, since the last 15 years, has become the most important part of molecular biology, because it's the forerunner of what we now call synthetic biology.
Yes, Khorana, who struggled a lot right from his childhood, Lahore, Multan, and then again struggling for the fellowship, and yes, you mentioned that how much he was struggling for his livelihood. And then lately he shifted to MIT in 1970, and he remained there for nearly 35 more years, remained active, nearly 35. It's little unusual for Kurana probably that to stay at a institution for 35 years. How do you see that, Dr. Sarkar, that he was traveling from one place to another, he was active throughout the North American region, United States, Canada and Europe. He has worked with Czechoslovakia and other places, and remaining and staying back at MIT for 35 years makes us little anxious to know. Can you throw some light?
Remember that by that time he had won a Nobel Prize, he was a Nobel laureate, he was one of the most respected people in the field, and he was given every facility that he wanted to have a large lab. And he was at a place which was easily becoming one of the world's great places to do molecular biology. Salvador Luria was there, Cyrus Leventhal was there, and all of it. And so I think at that point he had no intention of moving again, because notice that every time he moved from a place that was not as prestigious to a place that was better and more prestigious. And Wisconsin at that time was one of the world's centers for chemistry. By the time he got to MIT and he settled in, there was no place better than MIT that he could go to. Okay. I mean, he was at the height of its powers, he was surrounded by the Nobel laureates, he was well funded, and he liked living in Boston. His living situation earlier on in his life had not been very pleasant. He was an Indian. He was married to a Swiss woman, so it was an interracial couple.
Yes.
His family tells stories of him being called a Paki back when he was in Cambridge, or even when he was in Vancouver. When the family was in the United States, they would not dare travel to southern parts of the United States because they were an interracial family. In that context, settling down somewhere in the Northeast in a liberal part of the US made perfect sense for him. And he never moved again. And the family was very, very happy in Boston area, and Julia still lives there.
Yes, we can understand. Now, Dr. Sarkar, मन तो करता है आपसे बात करते रहें, खुराना साहब के बारे में बात जानते रहें, बट टाइम की एक लिमिट है. बट आई हैव डॉक्टर सुनीता फरकिया विद मी. शी हैज सम क्वेश्चन टू यू, सर, प्लीज. Sunita ji.
Dr. Sarkar, very quick, I will ask you one very small question, that what motivated Dr. Khorana for his great work? However, you have unveiled all the life about the great scientist, very small things about the great scientist. I just want to know for our viewers that what motivated Dr. Khorana for his great work? Thank you.
Kurana did not leave behind very much personal writing at all. He liked to talk science and science at all. My own sense of talking to him a few times and reading between all of this is that he was firmly committed to the idea that you could give a material explanation of all living phenomena. And he was doing in the laboratory what materialists had said for a long time could be done, but never quite accomplished. I sometimes wonder as to whether or not this is connected with the fact that as a young man, he went out of his way to go to a meeting, international meeting for young people and students organized by the Soviet Union. But this is speculation, and we cannot absolutely show with any real confidence what personally motivated him to that extent. But he was driven. He was going to synthesize living phenomena outside the living organism in the test tube, and he would do it step by step by step, no matter how difficult it is. And that's why when it comes to neurobiology, he chewed on possibly the most difficult of these problems, and those, as he knew by the end of his life, would be things that would require many more years than he had for their solution.
So, Dr. Sarkar, yes, we thank you very much for today's talk and today's coming to us and being with us. This is indeed our effort in bringing Indian diaspora to the motherland. Dr. Sarkar, we will be talking to you again. You have already left some thread for the next talk with us on this forum, that's on the first synthesis of a complete functional gene, etc. And these are besides the other brilliant achievements of Dr. Khorana and then this genetic code. So we really thank you very much for being with us, and we indeed thank all others, those who have helped you in bringing out this talk to us. And we really look forward for your new forthcoming book on Har Gobind Khorana. We thank Julia, Marvin, Binny Joseph, Raj Bhandri, Thomal, Saul and Matthews. Thank you all of you, my dear friends. Thank you. Looking forward meeting you again, Dr. Sarkar, on this forum. Thank you very much.
Thank you very much for having me. It was a lot of fun. Thank you.
Thank you. So, viewers, I hope all of you have enjoyed today's deliberation by Dr. Sarkar on Har Gobind Khorana. So, next time we are again here on the same platform on Tuesday. Now, we are going to have a talk by Anil Bharadwaj. He is the Director of Physical Research Laboratory, Ahmedabad. This laboratory is a laboratory of Indian Space Research Organization, ISRO, and has been doing pioneering work in space missions. So, Bharadwaj will be talking on Indian planetary missions to all of you. If you have any question, you can still send us all those questions to on our email addresses which we had given before. You can give us telephone calls for those questions. We will welcome if your suggestions for the talks to be held in recent future. आगे से आप किसी सब्जेक्ट पर चाहते हो आप हमें ज़रूर बताइए, हम उन विषयों का समावेशन करने का पूरा प्रयास करेंगे। धन्यवाद। तो आप लोगों से फिर से मुलाकात होगी इस मंगलवार 16 तारीख को 16 नवंबर को सुबह 10 बजे। धन्यवाद।